Hi guys, and welcome to our module on the introduction to hematology. Hematology is the study of blood and its disorders. Now, blood is a connective tissue that is found within the intravascular space of the extracellular fluid compartment. As health workers, when faced with diagnosing a condition, we tend to take blood as samples and send them to the lab to run tests. This is to check for various parameters that can give us an idea as to the diagnosis of the condition we are dealing with. And that's what we really need to remember when focusing on the entire model of hematology. The fact that we use blood to diagnose different conditions. And before we can talk about those conditions, we need to understand what blood is, the various components of blood, and the various parameters that we can get from the sample of blood we take to the lab. Blood has numerous functions, including the function of transportation, of storage, of immunity, and of homeostasis. In terms of transportation, the hemoglobin present within the red blood cells are involved in transportation of oxygen and carbon dioxide necessary for respiration at the cellular level. Blood also is involved in carrying nutrients like glucose, hormones, and enzymes to various aspects of the body. Blood is involved in storage and immunity. In terms of immunity, white blood cells and blood is involved in fighting against germs, both external and internally, and providing protection against these various microorganisms. And then in terms of homeostasis, blood is involved in the regulation of water balance, of acid and base, and then of the body temperature. And these are the various functions that the blood is involved in doing, alongside some others that are not necessarily stated in this particular slide. So about the composition of blood. Blood is a connective tissue, and it is composed of a combination of extracellular matrix and cellular components. The extracellular matrix of blood is known as plasma, and then the cells found within this extracellular matrix includes red blood cells, also called erythrocytes, white blood cells, also called leukocytes, and platelets, also called thrombocytes. So this is just a breakdown of the composition of blood. Remember that the cellular aspect of blood is made up of three major types of cells: red blood cells, white blood cells, and platelets. And in the case of the extracellular matrix, also called plasma, plasma is combined into two parts. We have serum and fibrin. Serum is the fluid aspect of plasma that is without fibrin. Fibrin is a proteinous structure within plasma, separate from serum, is usually involved in the process of clotting. So whenever we have plasma being taken or blood being taken out, and the blood is allowed to clot, what happens is that blood loses fibrin, and the plasma in the blood becomes serum. So remember that plasma is a combination of serum and fibrin, and then cells present within the blood would be the red blood cells, white blood cells, and the platelets, and the combination of two of this, the cells and the plasma, makes up blood. So this is just a picture of the different compositions of blood. You can see the plasma above, containing proteins within it alongside fibrin. We could see just below platelets and white blood cell, and then red blood cell taking a large chunk of the cellular aspect of blood. Let's talk about each of the components of blood, starting with plasma. Plasma, like I said, is a fluid aspect of blood. It's a part where the blood cells are suspended in. It's the medium by which the blood cells are transported around the blood. And it's composed of a combination of water, 92% of plasma is water, plasma protein, which is about 7%, and a number of solutes. The distribution of cells within blood is not equal. Usually, red blood cells take the chunk of the distribution of proportion of cells. It's about 99.8% of cells within the blood. Red blood cells about 0.1%, and platelets about 0.1%. Red blood cells are also called erythrocytes. They are the most abundant cells in the blood. They contain a very important iron compound called hemoglobin, and this compound is involved in the transportation of oxygen through the blood. White blood cells are also called leukocytes. They are the cells that tend to be involved in the immune functions of the blood. They include the cells known as granulocytes, agranulocytes, and lymphocytes. So this is an image that shows for us the various subtypes of white blood cells, starting with the granulocyte. Granulocytes are cells that contain visible cytoplasmic granules. They include neutrophils, eosinophils, and basophils. The agranulocytes are also called monocytes. These are cells that are typically known as monocytes in the blood. They have no visible cytoplasmic granules under light microscopy. That is in contrast to granulocytes. Whenever a monocyte leaves the bloodstream and enters into the extracellular space, we refer to them as a macrophage. And in case of lymphocytes are cells that tend to be located within the blood, but could also be found in extracellular tissues, what we call lymphoid tissues, including tissues like the lymph nodes, like the thymus, like the spleen, and other sites. And the reason why we call them lymphocytes is because of that association with lymphoid tissues. Lymphocytes in cell in the blood tend to either be classified as a T cell or a B cell. This is an image of a neutrophil. Neutrophils are also called polymononuclear leukocytes, and that's because of the fact that they have multiple nuclei within their cytoplasm. You could see to count as many as 5 to 6 nucleus here. Usually they tend to have about 3 to 5 nuclei within them. Neutrophils makes up a large chunk of the entire white blood cell. They're about 70% or two-thirds of the circulating white blood cell. Their cytoplasm is packed with pale granules that contain lysosomal enzymes and usually tend to stain a little bit pinkish slash purplish. Usually has a neutral color. That's how we tend to remember them. And their major function is in the function of phagocytosis. Neutrophils are involved in the phagocytosis of particular microorganisms, especially bacteria. This is an image of an eosinophil. Eosinophil takes about 2 to 4% of the circulating white blood cell. They have granules that tend to stain with a special stain we call eosin, and they are particularly found in allergic reactions and parasitic infections. Usually, it's like cases or conditions where we see high amount of eosinophils being involved in those situations. The image on the slide is that of a basophil. Basophil makes up less than 1% of the circulating white blood cell. Their granules tend to stain with basic dye, as in contrast to the granules of the eosinophils. You can see the granules stain with bluish, and the granules tend to contain large amount of histamine. Um, Basophils within the blood are known as basophils, but whenever they leave the blood and to enter into the tissues, then we refer to them as mast cells. They discharge this histamine in situations of inflammation or allergic reaction, and usually in cases of allergies like asthma, we tend to see basophils and their mast cell counterparts in action. This is an image of a monocyte. Monocyte makes up about 2% to 8% of circulating white blood cell. What is unique about them is the fact that they are agranulocytic. They don't contain granules as shown in the image. And most importantly, their nucleus tend to be kidney shaped and this is one way to identify them in contrast to the other white blood cell we talked about. You can see the kidney shape of the nucleus of the monocytes. What you need to also remember about monocytes is the fact that when they leave the tissue, when they leave the blood vessels to enter into the tissue, we refer to them as macrophages. Macrophages, in terms of functions, are phagocytic. They are very similar to neutrophils in terms of their action, and they have various names in different sites wherever they are found. So in the skin, they are referred to as dendritic cells or Langerhans cells. In the brain, we know them as microglia cells. In the kidneys, we know them to be mesenteria cells. In the lungs, we refer to them as alveolar macrophages. The liver, we refer to them as coffer cells. In the spleen, we refer to them as histocytes. So they have various names at different tissue sites. But remember that macrophages are they are gotten from monocytes while the monocyte is has left the blood vessels and entered into the tissue. Finally, we have the lymphocyte. Lymphocyte makes up about 20% to 30% of circulating white blood cells. They are relatively small, slightly larger than the red blood cell, and they have a very large nucleus. So if you are to compare this to the other cells, the white blood cells, the nucleus of the lymphocyte is extremely large. It's almost taking 98% of the cytoplasm. And the lymphocytes are involved in various activities. The B lymphocytes, for example, or B cells in particular, are involved in the production of antibodies, whereas the T cells are involved in the fight against viral infections. So whenever there's a viral infection, we have more of T lymphocytes being in action. Whenever we have a bacterial infection, we have the B lymphocytes being involved. They are involved particularly in production of antibodies. Last blood cell are the platelets. Platelets are thrombocytes. They are blood cells involved in the process of blood clotting or hemostasis. Whenever we have a ruptured vessel or a damage to an endothelial surface and there's bleeding occurring at that site, platelets would usually gather, be activated, and then they will block the site of injury to prevent blood loss from occurring. Let's talk about the properties of blood. Whenever we take a blood sample, four important parameters we should always remember would be parameters of color, of volume, pH, and of specific gravity. The other parameters and other constituents of blood that we tend to test for, we talk about that when we talk about the other modules or when we get to the other modules under hematology. In terms of the color of blood, whenever we're taking an arterial blood, the blood tends to be scarlet red. Usually, we don't really tend to take arterial blood except maybe when we need to run various tests like a pH test or an arterial blood gas test. But most of the time, the type of blood sample we take tends to be venous blood. This is always purplish red or dark red in color. In males, the volume of blood is about 5 liters. In females, about 4.5. And there's presence of the pH of the pH of blood normally tends to be alkaline, usually about 7.4 in terms of pH, whereas the specific gravity tends to range from 1.052 to about 1.061. That concludes our module of the introduction to hematology.